In the world of microbiology, sometimes it's the simplest tests that have the biggest impact. Today, we're going to break down one of those cornerstones, the Coagulase test. This is pretty much our #1 tool for unmasking one of the most important bacterial pathogens out there, Staphylococcus aureus.
So, just picture this for a second. You're in the lab, a culture comes back, and it's positive for Staphylococcus. Now you've got this critical question on your hands. Are we dealing with a harmless bug that just lives on the skin, or is this the really nasty one, S. aureus, a culprit that can cause everything from skin infections to life-threatening sepsis? Well, telling them apart, and telling them apart fast, is absolutely vital for patient care. And that, right there, is where the Coagulase test comes into play.
So before we dive into the how of the test, let's really get a handle on the why. What is the clinical significance? Why is this one little test so incredibly crucial in our diagnostic toolbox?
To put it as simply as possible, this is the single most important biochemical test for telling Staphylococcus aureus apart from all the other staph species. Just think of it like a bright, clear dividing line. On one side, a major human pathogen, and on the other, its less aggressive relatives.
In this slide, it shows you that fundamental split perfectly. The test literally divides the entire Staphylococcus family into two major groups. You've got your coagulase-positive S. aureus on one side, and on the other side, all the others, which we lump together and call Coagulase-Negative Staphylococci, or CONS for short. Making this distinction is absolutely key for figuring out the right treatment.
Okay, so how does this whole thing work at the molecular level? Well, it all comes down to detecting the bacterium's secret weapon, this really powerful enzyme called coagulase.
Coagulase is this amazing little enzyme that basically hijacks our body's own clotting system. It takes fibrinogen, which is just a protein floating around in our blood, and turns it into these sticky, insoluble strands of fibrin. This forces the plasma to clot up. Now, during a real infection, this creates a sort of protective fibrin shield around the bacteria, letting them hide from our immune system's attack cells. Pretty sneaky, right?
So let's walk through that pathway really quick. First, the coagulase enzyme goes and activates something in the plasma called prothrombin. Then, this newly activated prothrombin acts kind of like a pair of scissors, snipping fibrinogen into those sticky fibrin strands. And finally, all those fibrin strands weave together to form a solid, stable clot, and that clot is exactly what we're looking for in the lab.
Now, here's something that's really crucial to understand. Staph aureus actually makes two different forms of this enzyme. First, you've got bound coagulase, which is often called clumping factor, and it's literally anchored to the bacterial cell wall. Then you have free coagulase, which is an enzyme the bacterium just squirts out into its environment. And because there are two forms, guess what? We have two different ways to test for them. So that brings us to our first method, the slide coagulase test. The best way to think about this one is as our rapid screening tool. It's fast, it's easy, and it gives us a quick preliminary answer.
And what is it looking for specifically? It's designed to detect that bound coagulase, the clumping factor that's stuck right on the surface of the bacteria.
The procedure itself, it couldn't be easier. You just take a little bit of a bacterial colony, mix it into a drop of saline on a glass slide until you have a nice, smooth, milky suspension. Then you add a loopful of plasma, give it a little mix for a few seconds, and you just watch very closely to see what happens next.
The results are basically immediate and super easy to see. A positive result looks like coarse, visible clumps forming within just a few seconds. That's the clumping factor on the bacteria literally grabbing onto the fibrinogen in the plasma. And a negative result? It's just nothing. The mixture stays perfectly smooth and milky.
Okay, but listen up. Here's a huge exam alert. And this is a really common pitfall. The slide test isn't foolproof. There are a few other coagulase-negative staph species, and the one you really need to remember is Staphylococcus lugdunensis, that can also produce this clumping factor. And that can give you a false positive result, which is exactly why any positive slide test absolutely has to be confirmed.
And that, of course, brings us to our confirmatory method, the tube Coagulase test. Now, this isn't just a simple screening tool. This is our definitive, gold standard test.
So why is this one the definitive test? Well, it's because it detects free coagulase. That's the enzyme the bacteria actually secretes. This is way more specific to Staphylococcus aureus, and it's why we rely on this test for the final say.
Now, the procedure here takes a little bit more time. You take a colony and you mix it directly into a test tube that has plasma in it. Then you pop that tube into an incubator set at 37 degrees Celsius, body temperature, and you come back to check on it in about 4 hours. Reading this result is also super straightforward. A positive test is the formation of a solid, gel-like clot. The real test is when you tilt the tube. If it's positive, the clot stays put, it won't flow at all. A negative result is just what you'd expect. The plasma is still completely liquid.
And here we go, our second really important exam alert. Don't be too quick to call a negative result. If you check at 4 hours and there's no clot, you're not done yet. Some strains of S. aureus are just slow coagulase producers, so standard protocol says you have to incubate the tube overnight and check it one last time before you can officially call it negative.
All right, let's pull all of that together and wrap this up. Here's the final summary. This is the key information you're going to need for diagnostics and, of course, for your exams.
This table right here, this is the ultimate takeaway. The bottom line is really this simple. A positive coagulase test means you've identified Staphylococcus aureus. All the other medically important staph you'll run into, like epidermidis, saprophyticus, hemolyticus, they are all coagulase negative. It's really that clear-cut.
And that's the Coagulase test, a simple, kind of elegant procedure that is still absolutely essential in the clinical microbiology lab for identifying a major pathogen. If you found this breakdown helpful, then please like, comment, and subscribe.